Hello there, my name is Pome, and I'm a second year PhD student doing computational and theoretical neuroscience at the Neuronal Dynamics group, where we study how complex patterns emerge in neuronal systems using simple mathematical models analyzable to a large extent. One of the major challenges in neuroscience is to understand what are the neuronal mechanisms that give rise to the large scale behavior of the brain that we can observe microscopically. For example, if we put electrodes on the scalp of a person in order to perform an EEG, we might see some fluctuations in the voltage readings which are indicative of different sorts of brain activity. We call these fluctuations brain oscillations, and they are usually thought to underlie some kind of phenomenon of neuronal synchrony. But exactly how and why that happens still remains an open question. What's more, if we were to actually open the skull of the person and look at what the individual neurons are doing, we would probably see something like this. These are the voltage traces depicting the spikes of the two major types of neurons we have in our brains, which are excitatory and inhibitory cells. And it really doesn't look much like there's any synchronization going on, much less any oscillation. And that is because we actually need to add up the activity of many neurons to see that there is indeed an oscillatory pattern going on in the population. This is what we call weak or sparse synchrony, because the neurons contribute very regularly to the overall rhythm, in contrast with what we would call strong synchrony, in which neurons fire regularly at each cycle. Understanding this dichotomy between the macroscopic oscillation and the sparse activity of the individual neurons is not easy. And in fact, scientists have proposed a number of factors that might help generate such states. Indeed, if we add these ingredients to our simulations of neural populations, we do get results that are very close to what we observe in real life. But we still lack an intuitive and simple understanding of this phenomenon that doesn't involve adding more complexity to an already incredibly complex system. In our research group, we have developed a very simple model that treats neurons as phase oscillators, capturing only their essential intrinsic properties. And we've studied the dynamics that emerge through the interaction between the excitatory and inhibitory neurons. As a result, we have been able to show that sparse synchrony emerges naturally in populations of such simplified models, which allows us to get a clear and intuitive view of the mechanisms that generate weak synchrony. In addition, our study suggests that the only necessary requisite for the generation of weak synchrony is the interaction between excitatory and inhibitory neurons. Although they may not be quantitatively accurate due to their simplicity, our results contribute to the mechanistic understanding of how the brain and cognition works in a qualitative sense, and they give us some insight on how something as small as a neuron can give rise to the complex and intricate behaviors of the nervous system. Thank you for listening.